honor of Professor Bhide, a great physicist, scientist, teacher, thinker, and to us, the members of his family, an equally dedicated family man. His birthday was on the 8th of August, and incidentally, Professor Madhukar Apte, an old associate of Professor Bhide and a key member of our organizing team, has brought to our notice some very interesting points about today's date. On this very day, in 1902, Paul Dirac, a British theoretical physicist who has contributed a lot in the development of quantum mechanics, was born. He was awarded the Nobel Prize in Physics in 1933. It was Dirac, in fact, in 1930, who had theoretically predicted the existence of the positron, which was later on experimentally verified by C.D. Anderson in 1932. Another Nobel laureate, born on the 8th of August in 1931, is also a British theoretical physicist, Sir Roger Penrose, who received this honor for his work in astrophysics. It is interesting to note that Svetlana Savitskaya, a Russian cosmonaut, who is the first woman to have walked in space, was also born on 8th August in 1948. And another renowned personality to share this birthday is a well-known organic chemist, Sir Jack Baldwin. So, on this same day, Professor V.G.Bhide was also born. And each year, the members of the Bhide family, VNIT Nagpur, Physics Promotion and Charitable Trust Nagpur, and the Maharashtra Academy of Sciences Nagpur chapter organize a lecture in his honor on a scientific topic which would also appeal to the general public. The last 2 years, the COVID-19 pandemic has unfortunately prevented us from holding this event physically, but we are back again this year, and very fittingly, we have as our chief guest and speaker someone who has been key in our fight against COVID, Dr. Santosh Narwade from the Serum Institute of India, Pune, where he is the Scientist Alliance Manager for the Covishield vaccine project. I think first of all, I request all of you to put your hands together for the exceptional minds at the SII who have come up with this vaccine in record time when faced with extraordinary circumstances. We thank you, Dr. Narwade, and your team for all the hard work that you have put in for all of mankind actually. Professor P.M. Padore, Director VNIT Nagpur, will preside over our function today. And sir, may I reiterate how indebted we have always been to the VNIT for their steadfast support of our lecture series. Thank you very much. So without further ado, we get going with the proceedings, and I invite all the dignitaries, our chief guest, Dr. Narwade, President of today's function, Dr. P.M. Padore, Professor V.K.Deshpande, Dr. Rashmi Uddanwadikar, and Dr. Vidula Kapre, to all please light the lamp to honor the memory of Professor V.G.Bhide and then go on to occupy their place on the dais. Our chief guest, Dr. Narwade, of course, as I mentioned, is from the Serum Institute of India, where he is the Scientist Alliance Manager for the Covishield vaccine. Professor P.M. Padore is the Director, VNIT Nagpur. Professor V.K.Deshpande is the Head of the Physics Department at VNIT and is the Vice President of the Maharashtra Academy of Sciences. Dr. Rashmi Uddanwadikar is the Professor at the Mechanical Engineering Department and is the Dean of Public Relations at VNIT Nagpur. And Dr. Vidula Kapre is the Consultant Anesthesiologist and Managing Director of Niti Clinics Nagpur and is the daughter of Professor Bhide. Thank you very much, and I invite all the dignitaries and guests to please take their seats on the dais. I request Priyanka to please welcome our chief guest, Dr. Santosh Narwade, with the bouquet. I request Priyanka to present a bouquet to Professor Padore, sir. May I call Priyanka again to present a bouquet to Professor V.K.Deshpande, sir. I request Meenal to welcome Dr. Rashmi Uddanwadi. And can you please also present a bouquet to Dr. Vidula Kamre. I now invite Professor V.K.Deshpande, Head of the Physics Department at VNIT and Vice President of the Maharashtra Academy of Sciences, to deliver the welcome address and also give a brief introduction of Professor V.G.Bhide to our audience. Thank you.
Honorable Dr. P.M. Padole, Director VNIT and President of today's function. Chief guest Dr. Santosh Narwade from Serum Institute of India, Pune. Dr. Vidula Kapre, Dr. Madan Kapre, members of Bhide family. Dr. Rashmi Uddanwadi, Associate Dean Public Relations, VNIT. Invited distinguished guests and my dear student friends. I extend a warm welcome to all of you for this Professor V.G.Bhide Memorial Lecture. This lecture is organized by Maharashtra Academy of Sciences, Nagpur chapter, VNIT Nagpur, Physics Promotion and Charitable Trust, Nagpur, and members of Bhide family. After a gap of 2 years, this is the 14th lecture in series which has been organized in Nagpur, and it's a matter of great pride for all of us from the organizing committee. The reputation of Professor V.G.Bhide Memorial Lecture is very high because of the excellent speakers who have delivered these lectures. The most remarkable feature of these lectures is that the audience is a class audience from all disciplines of science, engineering, technology, and humanities, and also from medical practitioner, medical field. We will rarely find this combination in the audience, but we are proud to have this class audience for this lecture. In fact, the Nagpurians look forward to this V.G.Bhide Memorial Lecture very anxiously. I would like to place on record our appreciation and thanks to all those who love this lecture. This lecture has become an important event in the scientific life of Nagpur. Some of the prominent lectures recently delivered under Bhide Memorial Lecture Series are, I'll just give you a glimpse of it. Dr. Sanjeev Galande, Dean and Professor at IISER Pune, he spoke on nature versus nurture, how environment shapes our genes. Then we had lecture by Dr. Suresh Naik, ex-Group Director ISRO, on Chandrayaan-1, Mangalyaan and beyond. We had lecture by Dr. Sachin Deshmukh, Neuroscience Department of IISc Bangalore, on the topic perception, emotion, memory, how brain does it all. Then Padma Shri Professor K.L. Chopra, ex-Director of IIT Kharagpur, he gave a lecture on nurturing innovations and entrepreneurship in academic institutions. Professor Rajiv Dusane of IIT Bombay gave a talk on energy harvesting using solar energy. Then we had Dr. Brijesh Dikshit, Managing Director of Maharashtra Metro Rail Corporation Limited, who spoke on thinking big, building metro systems in Nagpur, a challenge in management. I still remember when he gave this lecture, there was just the metro work had begun in Nagpur, and we see the metro running today. And that time he shared a lot of things with us in this lecture. Then the last lecture of this series was given by Dr. Satyajit Rath from IISER Pune on immunotherapy and effective way to fight cancer. The topic of these lectures and the distinguished speakers give us an idea about the quality of these lectures, which were highly appreciated by everyone. As you would agree with me that the whole world was badly affected by COVID-19 for last more than 2 years. In fact, many of us have lost our near and dear ones due to COVID-19. We have lost our Honorable Chairman, Board of Governors of VNIT, Shri Vishramji Jamdar, due to COVID-19 in September 2020. Shri Jamdar had attended several lectures and presided over 12th and 13th Bhide Memorial Lecture in this very hall as a Chairman Board of Governors of VNIT. And he had expressed his great appreciation for the high quality lectures organized so far. All of us are aware that the Serum Institute Pune had played a very important role in the manufacturing of Covishield vaccine for COVID-19 on a large scale. This has helped not only India but people from several other countries during the pandemic in fight against COVID-19. We are very glad that today we have Dr. Santosh Narwade, a scientist, senior scientist and Alliance Manager Covishield vaccine from Serum Institute of India as a chief guest to deliver the 14th Prof. V.G.Bhide Memorial Lecture on novel strategies to combat COVID-19. On behalf of all the organizers, I extend a warm welcome to you, Dr. Narwade, and all those who are present in this hall. I am sure all of us are going to enjoy your lecture. For the benefit of new distinguished members of audience and our young students, I would like to take this opportunity to give you a brief introduction of Professor V.G.Bhide, in whose memory these lectures are organized. Professor Vishnu Ganesh Bhide was born in Nagpur in 1925. He obtained his master's degree in physics from Nagpur University with first rank and gold medal, and secured a doctoral degree from the same university. He started his career at the Institute of Science Nagpur in 1956 as a faculty of physics. Then he went to UK to study solid state physics and obtained doctoral degree from University of London. After his return from UK, he worked as the head of the Department of Physics at the Institute of Science Bombay. During this period, he initiated research on Mossbar spectroscopy, a pioneering effort in India. After serving as a scientific advisor to the Government of India from 1973 to 75, he moved to National Physical Laboratory, which is popularly known as NPL Delhi, as a director grade scientist. In 1982, he shifted to Pune as a chairman of School of Energy Studies and worked there till 1984, when he was appointed as a vice chancellor of Pune University. In 1988, he worked as a founder director of Inter-University Consortium for DAE facilities. Professor Bhide published several books and scientific papers in national and international journals of repute. He is known for his contribution to the field of science education in India. As the chairman of the physics study group, he was involved with the preparation of secondary school textbooks for the NCERT, that is National Council of Educational Research and Training. He served as the chairman of Asian Working Group on Solar Energy, as the director of International Solar Energy Society, and as a member of International Commission on the application of Mossbar effect. He was the president of Indian Science Congress in 1971 and served the Indian National Science Academy as its secretary from 71 to 74 and its treasurer from 81 to 84. Professor Bhide was elected as a fellow of Indian Academy of Sciences, National Academy of Sciences India, Indian National Science Academy, Maharashtra Academy of Sciences, Indian Cryogenic Council and Royal Astronomical Society. The Government of India awarded him the civilian honour of Padma Shri in 1992. He was also a recipient of Sir C.V. Raman Award and Meghnad Saha Memorial Award. Professor Bhide died on 25th June 2006. He was a great teacher, a scientist par excellence, an able administrator. He worked all through his life with a passion for science education and the popularization of science among the students particularly. We feel extremely proud and privileged to be associated with the prestigious Professor V.G.Bhide Memorial Lecture, and I welcome once again all of you. Thank you very much.
Thank you, Professor Deshpande. Professor V.G.Bhide, our Azoba, was indeed an exceptional man with many virtues, and his life's work and memory continues to inspire us all even today. There is a slight change in our agenda for the day. Our president for today's function, Professor Padore sir, will have to leave early unfortunately due to some important engagements. So may I first request Dr. Rashmi Uddanwadikar, Professor Mechanical Engineering Department and Dean Public Relations, VNIT, to introduce him and then invite him for the presidential address.
Thank you, Gauri. Honorable Director, Dr. Padore, speakers for today's V.G.Bhide Memorial Lecture Series, Dr. Narwade, Dr. Deshpande sir, Dr. Kapre Madam, all the invited guests and my dear students sitting over here. It's a privilege for me to introduce my own director, my own PhD guide and friend and philosopher. Dr. P.M. Padore, a diligent researcher and administrator who strictly follows morals and ethics, the director of VNIT Nagpur, is focused on strategic vision and long-term mission of VNIT Nagpur. Professor Padore's ability to comprehend the broad picture and think proactively enabled him to operate effectively in a variety of administrative roles at VNIT, including Dean Faculty Welfare, Head of Mechanical Engineering Department, Training and Placement Officer, Professor in Charge Alumni Affairs, and many more. Professor Padore joined VNIT's former VRC Nagpur in 1988 as faculty in the Department of Mechanical Engineering. Since then, he is involved in teaching, research and administration. Professor Padore is a vibrant personality, eloquent teacher, popular professor and a prominent researcher with a vision of using science and technology for benefit of society. Professor Padore earned his bachelor's in mechanical engineering from VRC and master's in machine design from IIT Mumbai. He completed his PhD from VRC Nagpur. Professor Padore works in the field of mechanisms and machine design, finite element method and biomedical engineering. He has guided 17 plus PhD scholars, 50 plus MTech students. He has 4 patents and more than 160 research publications in reputed international and national journals to his credit. He has visited various countries like UK, USA, Germany, Romania, France, Hong Kong and many more for presenting his research work. He has chaired technical sessions at prestigious international conferences, completed more than 16 R&D research projects in the area of appropriate technology, finite element analysis, funded by Department of Science and Technology, Ministry of Education, Khadi and Village Industries Commission, and All India Council for Technical Education. Professor Padore developed functional and appropriate technologies for rural sector to improve its livelihood. He is working as the president of Biomedical Engineering Forum and has also developed medical implants and other gadgets for medical fraternity. He is passionate about his research. He has numerous awards to his credit. To mention few of them, he is a recipient of Academician Par Excellence Award from Institute of Engineers. He has received Best Science and Technology Innovation Award from Government of India for KVIC sponsored projects. Professor Padore, a jestful personality, leads his team in a friendly manner. He is currently also handling the post of In-Charge Director of NIT Andhra Pradesh. Apart from achieving excellence in his profession, Professor Padore is a versatile personality, equally good at painting, sports and singing. We are honored to have you, sir, and would like to listen to your presidential address. Sir.
Thank you, Rashmi, for such a long introduction and very high words for me. Maybe because she is my PhD student. I don't know whether I deserve this. Good afternoon to all of you. Respected Dr. Santosh Narwade from Serum Institute, who will be delivering today's V.G.Bhide Memorial Lecture. My friend Professor V.K.Deshpande, Dr. Vidula Kapre, Rashmi Uddanwadikar, Dr. Madan Kapre, all the doctors, particularly in the field of medical sciences, because I think good number of doctors are here from medical fraternity and my own doctors from engineering and science fraternity. All the deans, heads of the department, faculty members, ladies and gentlemen. It's my pleasure and honor to welcome you to the VNIT for this prestigious V.G.Bhide Memorial Lecture, which all of us know that it is held on 8th of the August in the memory of Dr. V.G.Bhide on his birthday. And this is organized by Maharashtra Academy of Sciences, Nagpur chapter, then Physics Promotion and Charitable Trust, and the members of Bhide family from last many years. And I'm happy that from last few years, VNIT is also associated in organizing this event, and the credit goes to my friend Dr. V.K.Deshpande. So I must appreciate his effort that now we are also part of this V.G.Bhide Memorial Lecture. And I also thank members of the Bhide family for giving us this opportunity to organize this event at VNIT. It is a prestige for all of us. As all of us know that this event is a prestigious for this VNIT, and Professor Bhide was one of the best sort of a teacher, the researcher and administrator, and he has got a strong association with Nagpur. He did, as Professor Deshpande said, that he was born here and he did his MSc and PhD from Nagpur University, and he started his career from Nagpur and then went to Mumbai Institute of Science, and then as an advisor to the ministry at Delhi, and finally came back to Pune and became the Vice Chancellor of Pune University. So it is having a very bright career, but what is more important is that he spent whole of his life for promoting and popularizing the science among the students and also creating a scientific temper among the students. And that is very much necessary, I feel, because I know that when we were students in the schools, these science subjects and mathematics were supposed to be very tough for most of our friends. It was like a dragon or demon for all of us. And that is true because that time, whatever mechanisms were there for teaching, they were limited. It was just from the books. Very limited experimentation was possible. But now all of us know that from the government side also, from the institutions, from the various organizations, the efforts are made to make the science, you can say, the students friendly. A lot of gadgets are available. There are now audio, video, films are there, animations are there, games are there. And that is how we are trying to promote and popularize the science among the students. As far as Nagpur is concerned, we are all aware that Rajendra Singh Science Exploratory is there, then we have got Raman Science Center, then various adult tinkering labs are there at various institutions. So these are the things which I suppose we are trying to make the students friendly science, and then also trying to encourage the students to go for science. As far as VNIT is concerned, we are also making a deliberate effort to reach to the students, particularly for promoting the science under the Unnat Bharat Abhiyan, and also the Prayas Club. Today we are having the inauguration of that. We are reaching, our students and faculty members are reaching to the various schools and the students, and we are organizing various events for the school students at Nagpur, in the Nagpur at VNIT, like which is essays, then organizing the various science exhibitions, and that is how we are also trying to reach to the students. Every year, more than 2-3 such events are organized at VNIT. Then we also organized a event called Kutuhal last 2 years back, and I'm happy to share with you more than 25 students from the various schools. They visited that exhibitions, and just now we also organized Vigyan Sarvatra Pujyate. That event was also organized about 2 months back, and various students from various schools, they also visited that. So this is sort of a contribution from VNIT to promote and popularize the science among the students. As far as this lecture is concerned, as Professor Deshpande said, that this is a sort of a prestigious event for us, and it has set different standards, very high standards as far as lecture is concerned, and the prominent personalities from the various areas, they have given lecture over here, which includes Padma Vibhushan Anil Kakodkar, then Dr. Chopra, who was director of IIT Kharagpur, Brijesh Dikshit from Maha Metro, and Dr. Rath from Azhar Pune, and many such lectures. And today I am happy that in this series, Dr. Narwade will be delivering a lecture related to how to combat the situations of COVID-19. We are all, in fact, knowing how difficult these 2 years were, and we have faced the unprecedented situations in the last 2 years because of COVID-19. And as a director, I remember that I think on 15th of March 2020, I have to ask my students to vacate the hostels, and then we have to give our hostels for the isolations and other kind of things. So we faced a lot of problems during that, all of us in fact. But the organizations like Serum Institute, they came to our rescue, I can say, and in a very short period they could develop the vaccines. And it is not only that they have developed the vaccines in a very short time, but more than 200 vaccines, COVID vaccines doses were given to the Indian population. And not only that, they also helped the other countries by supplying the vaccines. So this is a great work that is done by Serum Institute. So I must congratulate the Serum Institute for doing such a good work for mankind. In this regard also, the VNIT has done something, some small research and development to help the society in and around Nagpur. I am happy to share with you that we have developed what is called as two-way ventilator splitter, because that time there was scarcity of the ventilators. We developed that, and one such ventilator splitter was in fact tested at IGMC, and it was found to be working rightly. Similarly, we also developed the Sahayak Robo for serving the patients in the isolation ward. It was having audio visual system, and it could serve the foods and the medicine to the isolation ward, and the patient sitting in the isolation ward was in a position to contact with the doctors who is sitting in the cabin. So this was also given to AIMS. Then we also developed UV based sanitizer for sanitizing the PPE kits and other kind of the things. So this is sort of a work that R&D work that we have done at VNIT. Then VNIT Alumni Association also did a lot of work for serving the NGOs and other things. We provided the oxygen cylinders, oxygen concentrators and other things in that during that difficult period. So these are the things that we did, and I am sure that now whatever lectures we will be having, that will be giving more idea how to in fact combat the situations of COVID-19, which all of us should pray God that that should not come, but we should be prepared for that kind of the eventuality. And I am sure that this lecture by Dr. Narwade will help us in future if this kind of situation arises. So once again, I welcome you all for this lecture, and thank the Bhide family for again giving us opportunity to organize this lecture at VNIT. Thank you.
Thank you, Professor Padore. As you mentioned, Dr. Bhide was very keen in his interest of developing curiosity in students about the sciences and popularizing science, and given all the projects that the VNIT is doing, I think this association of the Bhide Memorial Lecture with VNIT indeed is become more pleasurable. Thank you so much, sir. May I request Dr. Bhide to please present a memento to Professor Padore as a token of our thanks. I now request Dr. Rashmi Uddanwadekar to introduce our chief guest for today and then invite him for his talk. Thank you.
Thank you. This is such a matter of pride to introduce Dr. Narwade over here. He had a 5-page long biodata and it was a tough time for me to reduce it and cut it short to 1.5 page. I'm sure I have not missed out the most important and the highlight points. Dr. Santosh Narwade is having 13 years of research experience, is presently working at Serum Institute of India, Pune as a senior scientist and leading different projects from design, cloning process development, clinical material to commercial manufacturing. He has successfully handled Oxford AstraZeneca vaccine process, analytical development, scale up from 10 liters to 4 kiloliter scale, and delivered around 1.9 billion doses. So this needs a huge round of applause. So 1.9 billion doses worldwide with the brand name Covishield. He has developed Oxford candidate vaccine from tox, clinical trials to commercial product in a record time. Then process analytical development and scale up of Sputnik Light vaccine at 2 kiloliter scale. Sir, is that 2 kiloliters? Yeah. Designed a novel vaccine technology for COVID-19 vaccine and other respiratory diseases for which he has filed the patent. He has to his credit, clinical development for phase 1, phase 2 and phase 3 of quadrivalent HPV vaccine, Tdap vaccine, malaria plus matrix vaccine and many more very difficult names. First time designed and implemented single-use system throughout process from bioreactors up to fill finish. He is also looking after regulatory, clinical and marketing authorization dossier submission. He has many new projects in which he is discussing with European Union, US universities and other institutions. He has implemented new design projects at EU manufacturing site. He has established TEM, cryo-EM, NGS based methods for additional characterization, vector and protein based vaccines. Development of mRNA based vaccines and therapeutics. He has worked as an ICMR senior research fellow in University of Pune. Then he also worked as a junior research fellow in Molecular Biology Research Laboratory at Pune. Worked as research assistant at Department of Molecular Virology at Pune. And process development scientist at Genova Biopharmaceuticals Pune. He is MSc in Biotechnology and PhD in Biotechnology from University of Pune and JNU Delhi. He has 12 publications, 1 patent is granted, 1 is under process, and he is serving as a member of Board of Studies at different universities. The major highlight is he was highlighted in the New York Times for Covishield vaccine development in a record time. So we are very honored to have you, sir. May I request you to address our topic. Thank you.
So, thank you all for this nice program and thank you Vidya Madam for the invite for this program, today's program. Because actually I have a lot of pressure when I received the invitation for this Professor Bhide sir's memorial lecture, because I have just heard about sir because I have done my PhD from the Pune University. At that time he was served as a good Vice Chancellor for Pune University, and my PhD guy was recruited by Bhide sir. So that is one correlation with that. Yeah. So that for that actually I was happy, and he was also associated with the Serum Institute of India. So some projects I think they have done, and some funding also given by the Serum Institute of India for the Professor Bhide sir. And thank you. Dr. Madam for this nice introduction. So actually I don't know this is biodata is for the reading, so I have just mentioned my CV over there, so else I will give a small paragraph for that. And thank you sir, Madan sir, for this invitation, and Honorable president of this today's event, Dr. Pradwade sir and Dr. Deshpande sir from the Maharashtra Academy of Sciences, Nagpur chapter. So please bear, actually I am not good talker or actually I am just a researcher, but I will try to present my whatever the work, because as today I was expected to talk about the Covishield vaccine, but due to some conflicts and some issues in the public, and some confidentiality, so I have kept my topic open, that is the novel strategies for to combat the COVID-19. So these includes the diagnostic strategies, and also the therapeutic and vaccine, which types of vaccines, which kind of diagnosis actually we have implemented during this COVID pandemic situation. So just for understanding, how many know the biologically? So COVID is actually, everybody is now doctor. So everybody know about what is the COVID, what is the treatment for that, and everybody self-medicate itself to combat the symptoms of the COVID. So can I talk in the routine scientifically, or just I have to explain the background for that? What is the DNA and everything? So that I want to understand, or else I continue. Yeah. So now everybody know about the COVID-19. It is caused by the SARS-CoV virus, and having the symptoms like the fever, cough, fatigue, shortness of breath, and loss of smell and taste. And but actually these are the symptoms, but maximally around 80% of patients who has the exposure to the COVID-19, they are asymptomatic. On only the 15%, around the 15% people who has the mild symptoms, and only the initially in the Delta wave and the initial first phase of the COVID pandemic, so only the 5% cases actually they need some support, ventilation and everything. But whatever the situation worldwide was that actually who has tested positive, it is treated like that actually he has done something wrong to the society. So due to that actually we need to aware about which kind of diagnostics, and actually whether these tests are accurate or not, and how we have to predict the data. So that I will going to talk about that. So this transmission, so everybody know about that. So as of now around 58.2 crore cases of COVID-19 are detected and recorded worldwide, and during this Omicron wave, so maximum cases are non-detected. So everybody has tested at home, so this data is not included. And in the outside India also, nobody care about the recording the data now. But whatever the recorded data is there, so around 58.2 crore population worldwide get infected, and among that around 64 lakh people died globally. And from that, in India around 44 million cases are detected, and around 5 lakh death is there. First we start with the structure of COVID virus. So this COVID is enveloped virus. So there are two types of viruses. One virus is enveloped virus, other are only the capsid viruses, non-enveloped virus. So it has only the capsid outside, and the enveloped virus, the outside envelope will be there. So this is enveloped virus having the RNA, single-stranded RNA as a genetic material. So there are two types of genetic material, DNA and RNA. So in the RNA is a single-stranded and double-stranded RNA. So this is a single-stranded RNA virus having genome size of 30 kb. So if we sequence it, so it is around the 30 kb long genome size, and it is composed of the 4 major structural proteins. So whatever the different proteins available in our body, similarly virus has the different proteins. So among that, 4 are the major proteins, which includes the membrane proteins, and it is yes, these are 4 types of membrane proteins, which includes the envelope protein, so which is the outside covering of the virus. It is called as envelope proteins, and that whatever the support to the envelope protein is the membrane protein. So these two are the structural important proteins. Then the virus has the spike protein and nucleocapsid. Nucleocapsid is the covering, inside covering of the virus, virus nuclear genome. So it is the single stranded RNA, it is covered by the nucleocapsid, and the important therapeutically and the diagnostically important protein is the spike protein. So the spike protein, which is involved in the transmission or infection to the human cell line cells. And other around 16 are the non-structural proteins. These non-structural proteins are the mostly enzymes which are important for the viral replication and the virus assembly inside the human body. And these non-structural proteins like this RDRP, which is in the center. So this is one of the key enzyme used for the diagnosis of the COVID by the real-time PCR. Then the second one used for the diagnosis is the envelope protein. So this is E gene, this RDRP gene, these are mostly used. Then some cases nucleocapsid also used for this diagnostics in the real-time PCR, in the antigen kits also. And the interesting protein is this spike protein. So this spike protein which is important for the infection to the cells. And this virus is around tentatively around 65 to 120 nanometer size, but average whatever the matured virus is there, it is around 90 to 100 nanometer size. So this is because as the virus will mature, so size is different. And the spike protein, which is important, it is around 9 to 12 nanometer size. These are very tiny size virus particles, you know the virology. And for any disease, so when we have to come to the diagnostic and therapeutics, so in that case, we need to understand the genetic and the phenotypic structure or characteristics of these viruses, and which is important for this diagnosis and therapeutic. So for in this COVID, so when we look at the COVID-19 and its initial family, that is SARS, COVID and MERS viruses, so in that cases, the mostly non-structural proteins, these are conserved among this all these species, and the spike protein, which is normally target for the vaccine development, it is also conserved. Only some changes are there in the around 70% is the conserved regions. Conserved means similar to that, but the initially for the SARS and MERS, actually there is no commercial vaccine available. So if that vaccine is available, so there should be some protection in the initial days of the first phase of this COVID pandemic. And the main changes in these three types of families are the some structural protein, that is envelope protein, membrane protein, nucleocapsid. There is a lot of rearrangement and changes in the genome of this organism. Yeah. So when actually genetically, when we see the main target regions of this COVID-19, so it has different genes like the ORF, A, ORF B. So these ORF regions actually also targeted for the COVID-19 detection. One part of ORF is this RNA-dependent RNA polymerase. Then E and nucleocapsid is also the target. And the main therapeutic target is the spike gene. So this spike gene is divided into the S1 and S2, two parts. And this S1 part of the spike gene is called as RBD. RBD is the region, so which is the main part of this COVID virus, which binds to the cell receptor of the human cell line or any human lung cells and anywhere. So this is called as ACE2 receptor, where this RBD is get binded and the infection get progressed. So this RBD means a receptor binding domain. So which it binds to the ACE2 receptor, that is angiotensin converting enzymes. So actually, if there are actually once the virus infects the human body, so it starts the replication of its own replication. But during the replication, some viruses are unmatured. So unmatured viruses, they have close conformation of this spike protein. So whatever the spike protein present on these viruses, these are not open. So if it is not open, so it is not able to bind to the human ACE2 receptor. Virus is free, so actually there is no effect of virus on the human body. So for that some genetic mechanism is there. It is called as epigenetic mechanisms. So the methylation and the acetylation. So these are helpful for this spike protein opening. And if the spike protein is open in the open conformation, so in that case this RBD region of the spike protein get bind to the human ACE2 receptor and the infection get progress. So whatever the vaccines initially manufactured or designed, so that time they have not considered whether this spike should be open or closed conformation. So they have just take the whatever the sequence of protein sequence of the spike protein, they have designed the reverse engineer and designed the vaccines. But when actually we progress, means initial within 1.5 months, actually we come to know that actually this spike should be in the open conformation. For that actually we have made some different T2 mutations in that. So after that mutation, the spike is in the open conformation, like the flower, open flower. So that will be the one of the success for this Covishield also, and that leads to the higher immune response also against the specific regions, epitopes of the RBD. So now this is the background. So after that we will go to the diagnostic strategies used for this COVID-19, and many are the medical background, so they already know the what strategy they used for this COVID-19 detection. So why this is important? Because this COVID has whatever the symptoms shown by the COVID, these are the common flu-like symptoms. So we can't, on the basis of symptoms, we can't conclude that whether it is COVID or not. So in that case, so actually we need some confirmatory diagnostics test for that. And so the test can be the molecular diagnostics or serological test. So, but actually in India, if we consider the before COVID situation, so that time molecular diagnostics is around 1%, we can say. That also used for some genetic diseases and some HIV confirmation, some HIV viral load. Before that actually nobody has used molecular biology platforms for the diagnosis. The main limitation is we don't have the PCR systems or real-time PCR systems in the rural area also. In the Pune also only the few labs have this genetic testing platforms available, else only it is used for the research purpose. So for any diagnostic test, the specificity and the sensitivity of the test is important. The specificity means whether whatever the results given by the test, if it should be the detect the true positive itself. Else actually it gives a positive test to the negative, so its specificity is low. And the sensitivity is to identify the true negatives. Means the negative should be detected as a negative. In the condition, in the second wave, if someone gets the positive by the mistake, so in that case also it has a social impact, because our own actually family members also not going to get accepting us in the family. We have to quarantine. So for that purpose, the NAT testing and the serological testings are important. So for any testing, so actually initially concept-wise, so if some different companies start, so we have different approaches for the diagnostics. But when we progress to the some non-clinical samples, the clinical samples, but actually finally, if you consider we start development of 100 different types of concept for the designing of the kits. So in that case, we land up with the final 5 to 10 kits, which has good sensitivity and the specificity. So it is like any clinical trial. So similarly, it is the lab trial, but on the patient sample. So similar, this is the background for that. And in the nucleic acid testing, so first is everybody aware about the real-time PCR. So this is before COVID, only the research persons are actually aware of that, and some clinicians who are testing the viral load assays for the HIV. So this in the real-time PCR, there are small primers are there. So if somebody is non-biology and he is not understanding, so he can let me know, so I can explain in some common language. So please let me know, because many people are living.
Many are not doctors.
Huh? Many are not doctors. Yeah. So actually whatever the gene of target or gene of interest for the diagnosis. So against that, small primers are designed. Primers is around the small stretch of 20 base pair. Base pair is the bases are the ATGC. These are the bases of the any gene of the DNA actually. And for RNA, it is uracil instead of the thymine. So the small stretch of primer is used, and this primers has it is specific to the target gene, and with the help of the reverse transcriptase enzyme. So reverse transcriptase is the enzyme which is specifically available in the viruses. So this enzyme, so normally central dogma of the protein synthesis in that, so DNA forms the RNA forms the protein. But this viruses has the advantage, or it has some additional power, so it can convert the reverse to the whatever the central dogma is there. So for that, the specific enzyme called as reverse transcriptase is used, and my PhD boss was the first inventor of this reverse transcriptase at ALA University. But there actually the other person get the Nobel Prize for that. But this is the key enzyme used for this real-time PCR also, and it is the mechanism of viral escape from any disease for any treatments. So this reverse transcriptase enzyme convert this viral RNA to the cDNA, and this cDNA then using the normal PCR, using the DNA polymerase as enzyme, so it multiply itself and create a lot of copies of this DNA, and that can be detected by different methods. So in this real-time PCR, normally the fluorescence dye is used, and this fluorescence is captured. So during this COVID condition, so for the screening, E gene is used, for confirmation RDRP, N gene, and this ORF1b or ORF1a is used, and whether the swab sample is collected correctly or not. So that for that confirmation, one human gene, that is RNSP, is used. Else actually somebody has taken the outside swab and given for the testing. So that confirmation get by this RNSP genes, only whether the swab is from the human sample or not. And initially, when actually the first COVID kit was developed, so that was developed by my wife at my lab, Discovery Solutions. So she was working over there. So that time, so it is one gene is detected. So in that case, if actually in the PCR system we have to run, so there are different plate formats are used. So 96 plate format normally used for this PCR reaction. So but actually we have to use screening, confirmation, and internal controls. 3 genes we have to use for the diagnosis. Means in one run, in around 2.5 hours, so we can get only the 33 samples maximum. Because one sample we have to run in the 3 different ways. So she has multiplexed it, means in one well itself she has added 3 different genes, and that multiplex PCR will work, and it will give around more than 90 samples per well. And also that test can be pool testing, means actually if there is some exposure, but we have to detect everybody if somebody is. So in that case actually we can pool everybody's samples, 10 or 20 samples, and we can test. If these 20 are negative, so in that case actually there is no repeat testing required. If somebody is positive from this 20, then we have to individually detect this 20. So that approach also possible by this method. Only problem during the first wave is that actually there is no installations of the RT-PCR systems. So RT-PCR is not there, so actually that was the major limitations. But after 6 months time, so actually every district also get the RT-PCR installation. And other problem was this system is that actually different temperature changes are required, because the temperature cycling process was there. So it took a lot of time, around 1.5 hour for this main PCR reaction. So that was one limitation for this. So to overcome that limitation, so another method is used, that is called RT-LAM. So it is similar to the RT-PCR, but actually it is carried out at the single temperature, that is 64. In the RT-PCR we have to go, there are around 35 to 40 cycles are there. So for each cycle we have to 65, 90, then we have to cool down to the 4. So these cycles are continuously going. So, but in this case, and that required the trained staff to handle this RNA and everything. So this is the easy one method, but that only we have to take the swab sample, we have to mix in the tube, and we have to load that sample in the cassette, and cassette or tube we can use. So this tube can be incubated at 65 degree for 15 minutes, and it has the dye. So this intercalating dyes are used. So these dyes can be visualized by using any spectrophotometer or any colorimetric reading. But actually this was a simple and sensitive method, but actually it is not readily get commercialized because the manufacturing problems. The problem is with for this single temperature amplification. So whatever the enzyme required, so this enzyme availability was also one of the problem. So initially, you might heard that actually Abbott has brought one kit which is around 15 minutes. It will give that is Cartral based kit. So it has used the similar technology. So whatever, but actually it has the specificity issues somehow. So actually it is not worldwide used. Then this is the latest technology where the CRISPR-Cas technology is used. So CRISPR-Cas will be the game-changing technology in the future, because if actually in the normal genetic engineering, so if we have to do some changes in the genome, so we have to do the mutations in that. So sometimes these mutations are not allowed for these therapeutic purposes. But this CRISPR-Cas technology, so in that actually we don't need to do the mutations. So there is Cas9 is enzyme, and these CRISPRs are the some identification marks in the genomes. Just we have to identify that mark, and this enzyme will replace, or we can add whatever the nucleotide we have to do there. So normal routine mutations are not required for this process. So similarly, this technique is also used for this diagnosis. Yeah. So this also developed by the Sherlock Biosciences, and it is also commercialized at few places, but it is not worldwide also accepted. So in the future, actually, this technology also helpful for the point of care diagnosis. So that is my thinking. So this is actually whatever the limitations of the CRISPR-Cas was there. So this technology, so amplicon detecting by the lateral flow. So in that CRISPR-Cas and the RT-LAMP, so both technologies are combined. So in that actually whatever the patient swab sample is collected, so we have to just mix inside into the tubes, and it is like the RT-PCR, it get amplified on the isothermal, one temperature, that is 62 degree, and then we have to keep this tube into the 37 degree, and we have to add the Cas9 and the Cas12 enzyme, and this after that this Cas10 enzyme, so it has some fluorophores on that. This fluorophores will be detached by this Cas12 enzyme, and this samples is loaded onto the lateral flow, and we will get the results. So this is also the point of care test at the rural area we can use. So it is as sensitive as the real-time PCR, and we can do on the table itself. So that is also one of the advantage. But actually in India, CSIR, IGIB has actually they have developed this technology, but they are also not able to commercialize. They have, I think, collaborated with the Tata, and at few hospitals actually they are used, but they have actually for any test the technology is different, the sensitivity and specificity is different. The sensitivity and specificity is the part of optimization of that manufacturer. So that is lacking in this test. But actually these tests are actually the easy one, and it can be implemented for the many diseases as a point of care testing. So this is the microarray technology. So microarray technology is the old technology, but actually it can be used or implemented for the COVID detection, specifically during this variant analysis. So whether this is a wild type or not. So for that, whatever the reference genome, so the variant we have to detect. So we can take the RNA from that, we can label it with the Psi3, Psi5 dyes, so the different color dye. And the similarly, the patient sample can be fluorescence label, and we have to just hybridize to this plates. These plates are the microarray chips are there, and they after hybridization we have to incubate and we have to just scan this plate. So it will give the whether the both the just reference sample and the patient samples, whether these are matching or not. So it will give the whether it is variant of which is matching to the reference sample or not. It also gives the gene expression. So all these dots are specific to the specific genes, okay? Whether these genes are expressing that viruses or not. So that can be also detected by using this. The game-changing technology is the NGS analysis. NGS is the next generation sequencing analysis. So if we add the swab sample, so it will give the complete genome sequence of this patient. So from that, initially people have used this NGS analysis in the COVID for this because in the initial days actually the variants are not the issue, because that time only the one variant was there. So whether the secondary infections, which kind of secondary infections are happening in the respiratory tract, so that is one curiosity. For that purpose, this amplicon sequencing with the metagenomics is used. So this amplicon sequencing, it will give the sequence of the virus, the COVID virus. With that, it will give the which kinds of bacteria are prominent in the respiratory tract or the nasal cavity. That also identified by this metagenomic approach, and that will be also helpful for inhibiting the secondary infections caused by this COVID-19, because maximum deaths of this COVID-19 are due to the secondary infections. So that was one problem, because everybody is on the mucormycosis is also the secondary infection. Everybody on the steroids, so the immunocompromised, in that case the other bacteria, the commensal organisms also attacking to the humans. So but nobody is trying to detect actual what happening inside this ICU, whether the doctor is approaching to the patient, nobody knows in that condition. And this I have used this amplicon sequencing for different variant screening. So whatever the variants are circulating, so delta variant, delta plus variant, the main Wuhan variant and the Omicron variant. So all variants and lineages actually I have tried after vaccination whether these are present in the actually after vaccination, what is the effect of this variant on the patient, whether they are getting symptoms or not, whether they are getting the exposure or not. So that analysis actually I have done around 1,000 people. Actually these are our employees itself, but actually I have done this analysis. So during my conclusion for that during this year before this Omicron situation, so around mostly more than 70% of the adults above 18 get vaccinated. But in that case, in the around 15th or 17th of the February, so that time everybody is taking that actually Omicron is circulated everywhere. So whatever the infections we are getting, the Omicron infections are there. No, there is no symptoms for that. And when actually I have done sequencing analysis of these people, so these are the delta plus positive. Maximum people are delta plus positive, and the delta plus, which is most deadly infection in this COVID pandemic. So, but actually due to the vaccine, this delta plus is like the Omicron. Anybody is not getting the infection because everybody has a high titer of antibody. So that is the thing. Vaccine help or neutralize the viruses, delta plus. After 25th of the February, then the actual Omicron cases are getting detected by the NGS analysis. Before that, actually delta plus was there. So, but we are thinking that now the Omicron is there, nobody is getting some symptoms or severity for that. Then the serological testings will be there. So serological testing. So in the serological testing, only from the blood samples, all the previous molecular testings are on the basis of the swab samples. So from swab samples we are detecting the viral genome, and in the serological testing, we are actually from the blood samples we can detect the antibody titers. In the first before start of vaccination, initially people are thinking that antibody titer is important to detect the serological detection of the COVID-19, because for the HIV, then hepatitis, so in that cases we can detect serological titer for the HIV, tridot and everything. So the same concept was applied by all manufacturers, and they are targeted for detection of the antibodies. But antibodies, in the antibodies, there are two types of antibody for the common people. So non-biotech people. So the one is IgM. So after exposure to the viral infection, so first defense mechanism we get is the IgM antibody, and after the 14 days of this initial infection, so the same thing happens with the vaccine also. Vaccine is also we are adding some antigen. So against that, after vaccination, so after how much day we get the protection? That was the initial question from everybody. So after 4 to 5 hours, we are getting the IgM titer in our body. So this IgM titer will give the initial defense mechanism. So it lasts up to the 12 to 14 days. But after 14 days, actually we are getting generation, our body get expression or generation of the IgG antibody. So it is more protective antibody, and it has a half-life of, the IgM has a half-life of around 12 to 14 days, and in some cases it is less than 10 days. But this IgG titer, so which is getting after infection or after vaccination, so it has half-life 3 months of life, and but it can last for up to 6 months with the reduction in the titer. But the memory cells which is generated by the vaccine or the whatever the infection exposure also, so that can last for few years time. But initial, then if we get the infection after 1 year's time, so in that case initially we can get some symptoms, but that will be the milder symptoms as compared to the non-vaccinated person. So using this ELISA, we can detect, there are two types of ELISA, indirect ELISA and sandwich ELISA. So in this indirect ELISA, we can detect the antibody titers in the laboratory, and this sandwich ELISA, we can detect the antigen present in the human swab samples. But this test require to done in the laboratory, because this is ELISA based, so equipment, ELISA equipment is required for that. So, but this is important one. But in the countries like India, so there is no good infrastructure. Medically, actually, we are excellent in any terms, but in the rural area, the diagnosis is lacking. So in that case, actually, we need the point of care diagnosis. So the lateral flow test, that is our cartridge based home test, actually, which is developed by my lab, which is called as Covisel. So many of us actually we have used this test at the laboratory, at the home. So it is a rapid test in that the antigen is SARS-CoV antigen. It is detected in the lateral flow device. So it is also the good. It is having the good sensitivity above 90%, and but actually it has some specificity issues. So whether if this antigen kits give the positive results, so in that case the person is confirmatory positive. But if the person get the negative, so in that case sometimes the person has symptoms and the test is negative. So in that case the real-time PCR is suggested to the patient, because it has some issues with the negative samples. So these are some limitations of the serological test. Serological test actually sometimes actually the person's is seronegative. Seronegative means they don't have the antibody, but the molecular test can detect it is a positive, because the antibody generation takes time. So this is one limitation. And negative molecular. So actually sometimes the molecular test is negative, the real-time PCR is negative, but it has the antibody positive, because it is due to the post-exposure titer. But actually initial days these are problems, means actually antibody is positive, it is real-time PCR is negative. So in that case the person is sometimes treated as a positive. So these are limitations, because we have to another limitation is this test should give the confirmatory positive as confirmatory positive. But if in the COVID the situation like COVID, if there is any false positive or false negative result is there, so that will affect to the entire family. And family of the person. So we will come to the, these are initially we have completed the diagnostics, so we will come to the some therapeutic approaches including the vaccine. Okay. So vaccines and acquired immunity. So vaccines is actually the biological molecule or the antigen or the part of the whatever the target organism, that is bacteria or viruses, which is get injected to the human body, human beings, and against which actually we will get the immune response. So that immune response is called as acquired immunity, because that we have acquired using the external factors like the vaccine. And the other immunity is the innate immunity, that we have some non-specific immunity against different kinds of bacteria and viruses, that is innate immunity. So vaccine is the adaptive or acquired immunity. So for actually for these vaccines, as I told, before this COVID pandemic, the SARS and MERS actually they don't have any commercial vaccines. There are some developmental vaccines, but that also not gone to this clinical trial. So in that case, we have done for talks and everything, for designing the target designing, everything we have done with these COVID viruses. And it actually the normal vaccine development tenure is around the 7 years tenure. What we started considering the clinical trial durations, the designing, the animal testing, whatever. But we have done this whatever the COVID in the COVID situation. The government agencies also help us for these approvals on the basis of the limited data. So that help us to design and develop and the commercialize vaccine within few months trial. So for this COVID, there are different types of vaccine platforms and technologies used. So some vaccines are component vaccines, some are the complete viral vaccines. So initial days for bacterial vaccines, also for the viral vaccines, the whole viral vaccines are used. And these whole viral vaccines are divided into two types. One is inactivated and one is live attenuated. So in the inactivated virus vaccines, so direct vaccines, consider the measles vaccine, rubella vaccine, the rabies vaccines. These are complete viruses. So these viruses are grown inside the laboratory and directly they have killed by using the chemical heat treatments. And these killed vaccines are, the inactivated vaccines are used for the vaccination purpose. The Covaxin vaccine, which actually many around 8% of the Indian population has get exposure to the Covaxin. So that is inactivated vaccine. So the major problem is this inactivated vaccine is we have to use some chemical like formaldehyde for the inactivation of this vaccine. And sometimes these viruses may skip the inactivation, because from the large scale manufacturing we are testing some small quantity of samples. And this small quantity of samples is like एका शाबुदाण्याच्या भरणीमध्ये मोहऱ्या टाकल्यानंतर असं होतं, मोहरी असं निघून जातील. त्याच्यामध्ये काही शाबुदाणे आपले असे बाहेर येऊ शकतात ज्यांना की मोहरी नाही त्याच्यामध्ये. त्यास like the same situation actually, this is actually the good example for that. So from that we can't get the chance to test each and every virus particles, because we are only testing small sample from that, whether it is inactivated or not. So there might chances of small escape from the inactivation that leads to the disease. The vaccinated persons sometimes actually we heard that actually after COVID vaccination he get positive. So that is the reason for that, and many regulatory authorities have raised point for that also. And another is the live attenuated vaccines. So live attenuated vaccines like the BCG vaccine. So in that the actually culturing of the virus or bacteria for a long time, that also attenuated, means actually weaken the viruses, the whatever the toxic genes of these viruses, virus or bacteria. So that get attenuated or weakened. So that is used as a vaccine. As a vaccine, the polio vaccine is live example of this live attenuated vaccine, and the IPV, the injectable polio vaccine. So that is inactivated vaccine. So other new these are the new generation of vaccines. So these are called as component vaccine. So first is the protein subunit vaccine. This protein subunit vaccine. So in that only the RBD region of the spike protein or only the spike protein is cloned inside the bacteria or any viral expression system, and in the large scale bioreactors actually it get manufactured or grown inside these bioreactors, and the actual target gene, that is spike gene or RBD, so it get purified after this process, and this is used as the vaccine candidate. But actually only that small protein is not immunogenic that much, so it requires some adjuvant with that. So traditionally aluminum hydroxide, aluminum phosphate gels are used as a adjuvant for that. So but actually in our case, actually in the serum we have developed RBD based or spike based vaccines. So one of the example is Novavax vaccine, that is under the brand name of Covax. So that we received authorization globally for the US and UK and many other countries. So but after that actually we received the authorization in India. Before that US has recognized that, and that is a good vaccine for children also, because there is no direct exposure to the any virus inside the body. That is the purified protein subunit used for as a vaccine. And the second is VLP. VLP means virus-like proteins. So these are also the proteins, but during the manufacturing itself, we give shape of virus to that proteins. So for COVID also, we have tried this vaccine. We have used hepatitis vaccine as a base or backbone for that. Outside that, on the hepatitis proteins, we have attached this RBD proteins, and that is used as the vaccine candidate, but it is in the clinical trial itself. Then the DNA-based vaccines, RNA-based vaccines. So for that, the genetic material, the DNA used as a immunogen for that. So these DNA or RNA goes inside the bodies and then produce the spike proteins. Then the replication deficient vaccine, so that is a Covishield vaccine. So in that case, actually, whatever the spike gene sequence we have identified for this vaccination, so that spike gene we have used, we have cloned inside the adenovirus, that is a chimp adenovector. So chimpanzee oriented adenovirus, because adenovirus also infect to us and cause the flu-like symptoms. So so in that case, actually we might have some antibody titer against these adenoviruses. To avoid that, in our vaccines we have used chimpanzee oriented adenoviruses, those infect the chimpanzee, so we don't have the immunity against that. So that is used as a vector for this our vaccine. And this vaccine is non-replicating, means we have genetically neutralized or removed a gene which is required for the replication. So it is replication deficient, means in the human body it can't multiply. And the last example is replicating viral vaccines. So but actually nobody is actually preferring this, because sometimes these are non-lethal viruses, but it can multiply inside body. So I also not recommended to go with the replicating virus. For any vaccine, so we have to go to the preclinical studies. So means preclinical means on the animal, actually we have to show that actually it is immunogenic. Then we expose to the animal to the SARS-CoV virus. Then that is challenge study. So in the challenge study also we show that actually it gives the protection after vaccination. Then only actually we will get the permission for the phase 1. So in the phase 1 is only for the safety. It is on the 50 to 100 people, but we have to show that actually after vaccination there is no adverse event. Forget about the immunity and antibody titer, just for the safety purpose. Then in the phase 2, on the basis of our lab data, so we design the doses for that. So how much doses required for the human being. So that dose determination and safety and immunogenicity was done in this phase 2 trial, and the phase 3 trial was mass exposure. So in that we can expose up to the 5 to 10,000 people, and from the data we determine the efficacy of the vaccine. But the conditions like virus like COVID, so in that case the efficacy doesn't matter, because efficacy means he is positive after vaccination or not. But what is important is the protection or the avoiding the hospitalization. So that was main target. For this spike protein, we have targeted this RBD region and the complete spike gene for vaccine purposes. So first I will show the innate immunity. So what happens, so once we get the exposure to the non-vaccinated person with the COVID viruses, so in that case body has already innate immunity, innate immune response. Means actually initial days we heard that if somebody has strong immunity, in that case they don't have the severity of the COVID, and those who have weak immunity or comorbid, they have the effect of the COVID or hospitalization for that. So similarly, after the exposure of any virus or bacteria, our innate immunity will secrete the different molecules which are that is interferons. So that will protect us, and these are also termed as the cytokine storm, which is we get after this COVID infection, and simultaneously these are actually the dangerous signals or the molecules secreted. These are cytokine storm, and this is the mechanism by which these macrophages engulf these viruses. But sometimes these macrophages are insufficient, or they are not able to engulf all the viruses which will get exposure. So this is just background for that. So this is similarly high. Those who have high immunity, they have low infection rate, and those who have low immunity, they have high infection rate or severity. And if these low immunity, immunocompromised persons, when get the trained immunity is the exposure. We have to train our body that actually this is the protein from the virus, COVID virus, and that can come inside your body. So in that case, actually we have to prepare that, we have to defend against that. So that is the training, means vaccine is the trained immunity. So if we give the vaccination to that person, so in that case this hyperinflammation and the severity can be actually controlled. So for after the first dose of vaccine and the or the first exposure to the COVID, so in that case the immune response, that is a primary challenge, or first dose or the first infection of the COVID-19. So in that case at a certain level actually we will get the immune response. After that it will get down. So but when it actually get down, so at that time actually we have to boost that. So for that reason actually we have increased our second dose duration to the 3 months. But some people are telling that actually due to the unavailability of vaccine, they have extended the second dose. So this is the reason for that, because we actually we have used adenovirus. So there might be some antibodies against adenovirus. So that antibodies also get lowered during this 3 months time. So when that time we do the second booster dose, so that will increase the antibody titer at a higher level. So similarly, the infected and uninfected persons and the trained immunity or vaccinated persons, what is the difference in the protection? So that I have shown there. So this is one of the vaccine. So it is used for the boosting the immunity. It is called as RBCG, that is the recombinant BCG vaccines. So there was also the news, or there those who get the BCG vaccination at childhood, so they are getting the protection in this COVID wave. And in India, after 1980s, everybody get the vaccination of the from the BCG. In between a few years, actually they have stopped due to some issues. But outside India, there is no vaccination, specifically in the Europe. So they have more mortality as compared to the Indian populations, because we are received good protection in the first wave as compared to the second wave. So that is the reason. So for that we have this is the first vaccine worldwide which has carried out a clinical trial against COVID. For this BCG, all the frontline workers, we have given this around 5,000 workers, frontline workers, doctors and associated staff, we have given this vaccination at different centers, and check whether they have get any severity or infection or exposure or protection. So from this study. So this is good vaccine, and it is the advanced version of BCG vaccine, and it is the next vaccine which we all will actually receive in coming years time, because we are targeting for the TB eradication. So one of the arm of this TB eradication is this BCG vaccine. So that we have already tested for the COVID also. Then the mRNA based vaccines. So whatever the US and Europe, actually they Pfizer and Moderna, they have done this mRNA based vaccine platform. They have developed that. And there are two kinds of mRNA technology. So in the mRNA, so the whatever the target gene, spike gene or RBD gene of this COVID-19, so that spike gene is the sequence for spike gene is synthesized and it is cloned inside the one plasmid, the bacterial plasmid, and that bacterial plasmid is then in the laboratory, actually in the manufacturing facility, that bacterial plasmid is converted into the linear plasmid, and that is amplified and create the mRNA, messenger RNA copies. So it has message to transcribe that DNA sequence into the spike protein, which is required for the vaccine. So, but in all these other technologies, we are using directly the proteins, or it is inside any vector. But in this case, actually, we are only using the necked mRNA to vaccinate the people. So there are two types of mRNA vaccines are used. One is non-replicating mRNA vaccine, and another is self-amplified mRNA vaccines. And this mRNA, our body or everywhere we have RNAs. RNAs are the enzymes which cleaves this mRNA. So these mRNAs are actually very delicate and sensitive. So we have to protect that mRNA from the human beings itself, or these RNAs. So for that, actually these mRNAs are protected inside the lipid nanoparticles. So it is actually it is formulated in such a way that this mRNA molecule will get entrapped inside the lipid molecule, and these small lipid molecules used as a vaccine. And once actually it enters in the human body, so in this non-replicating mRNA vaccine, so actually we have to add large number of this lipid encapsulated mRNA. So against that, actually it forms this protein. This protein will give, form this antibody generation. And this self-amplified mRNA vaccine, we require small quantity of mRNA vaccine or as a vaccine candidate, and inside our body it will get replicated and forms the antibody. So this is mRNA based platform. Then the DNA vaccine, similarly the DNA sequence from the COVID-19, so that is also encoded inside the plasmid, and that plasmid is directly used as a DNA vaccine. So that is also not recommended, because the Zydus Cadila has this candidate, but in that case actually we are directly adding the one plasmid inside human body. So that is not allowed. So we don't know whether it will get integrated inside the human genome or not. So that is one technology. So another technology is live attenuated viral vaccine. So in the live attenuated viral vaccine, so we have developed one live attenuated viral vaccine, but actually these are genetically, artificially synthesized reverse engineering. By using the reverse engineering, genetically synthesized vaccine was there. So that is called as a Codagenics vaccine. So that is also one safer version of the Covaxin, because in the Covaxin we have to inactivate that. So it is genetically, though the unwanted genes from this vaccine is already removed. So whatever the vaccine is there, it is immunogen. So that is also under clinical development from our side. In the COVID, after vaccination, so many peoples, or the older age peoples or the immunocompromised peoples, they are not getting the good antibody titer, because they are immunocompromised. So in that case, or somebody get the higher exposure of the virus, in that case he has some symptoms or he has to hospitalize, and we have to immediately control the viral count. So in that case we need ready-made antibody mixture for that. So for the immunocompromised specifically, and those who get the high viral load and the severe infection. So in that case the ready-made antibody cocktails which are available, which are actually available by different manufacturers. So one is this Regeneron COVID-2 neutralizing cocktail. It is developed by the Roche pharmaceutical. So it is also manufactured in the India by the Cipla. So that is also, it is costly one, but it can save the life in the unvaccinated or immunocompromised persons. Similarly, AstraZeneca has developed this EvoShield as a antibody cocktail. So these are not much used in the India, but those who have, actually it is a high cost, around 60 to 80,000 rupees per dose, required 2-3 doses. So this is used in the India, but it is not preferred. But these monoclonal antibodies are, sir told that actually last time Professor actually they have given lecture on the immunotherapy on the cancer, I think. So for this same immunotherapy, though these monoclonal antibodies are used in the large number. So same kind of monoclonal antibody is tocilizumab. So this tocilizumab is used in the first and second wave to control the cytokine storm, which is caused by this IL-6. IL-6, because this IL-6, this tocilizumab binds to the IL-6 receptor, and whatever the unwanted effects of this IL-6, so that pathway it will control. So it will be helpful for the controlling the cytokine storm, or the lowering down the level of this IL-6 from the body. And this is one of the therapy actually it is given in India, the plasma therapy. So that is also not recommended, because with this plasma we are transferring everybody from the patient's body. He has any other disease, he has, we don't know what exposure he has. So we are taking that plasma, why? Because we need this antibody. So that antibody is already now ready-made. So this plasma has already adverse events, because that person might have some different kinds of negative cytokines which are not required for our body. So that also get exposed. So that is avoided. So we have also developed one hyperimmunosera from the equine origin, because this tocilizumab also has a limitation in the manufacturing, and this monoclonal antibody cocktail was also not available. So in that case, in the first wave itself, we have developed this RBD or anti-spike protein antibodies from the in the actually horses. We have generated antibody in the horses, and that purified and that used for the treatment of the patient. So it is also under clinical trial, but it has saved many lives. This molecule also, this heparin equine serum, because in the second wave, when actually we are not getting the bed, ventilator is long, actually that is a different aspect, but not getting the bed. So in that case, the patient get recovered at home by using this heparin serum. So now I will come to the whatever the topic of interest, that is the Covishield. So the Covishield is actually, I have avoiding to talk more about that, because there are some confidentiality and other issues with that. So this Covishield vaccine, so it is, as I told, that it is a chimpanzee oriented adenovirus. So it is a live adenovirus, but actually we have removed the E1 gene from that. So that E1 gene is important for the replication of this virus, adenovirus. And it is from the chimpanzee origin, so actually we don't have previous exposure to that. So that virus is used for the as a vector for this Covishield, and in that the spike gene was gene coding the spike gene was cloned inside that. So this is the chimpanzee. So we have modified it so that actually it will not replicate inside the human body after vaccination. Similarly, we have taken out the spike gene from this SARS-CoV-2 Wuhan variant, and that is sequenced, then the reverse engineer, and that sequence was actually used for cloning inside this adenovirus. So it forms this ChAdOx N COVID-19 vaccine, and when we vaccinate to the people, so it will express the spike gene inside our body, and it will against that spike, that spike gene goes outside the cells and they represent to our immune system, and our immune system will trigger the antibody response against that, and we are getting the protection from the COVID with this antibody, with this technology. So this is the adenovirus, this is the plasmid in which actually we have cloned this spike gene. That spike gene, this plasmid actually, by using the recombinant DNA technology, this the spike gene sequence and some other sequences which are required to express the spike gene inside the human body. So these are also added inside this adenovirus, and that is used as vaccine. And for this vaccine manufacturing, so whatever 1.9 million doses we have supplied or we have manufactured at Serum Institute of India. So for that, we have this. We have to grow the cells because this virus need the host, and for COVID, host is the human cells, and means actually we need to produce the vaccines which is humanized, means which is already grown or habituated inside the human cells. So for that actually we have used HK cell line, that is human embryonic kidney 293 cell line. So it is specialized cell line in which, but we have added this E1 gene which is removed from this whatever the adenovirus. So that gene we have added inside this HK293 cells. Means this virus is not replicating inside human body, but it can replicate inside this HK293 cells. So this is some novelty of this technique. So but actually before manufacturing large scale vaccines, we have to grow these cells first. So from 1 ml of the cells, we are multiplying these cells inside the laboratory or inside our manufacturing facility to the 2000 liter, 2 kl or 4 kl. So I have just represented up to the 2 kl. So we have grown up to the 6 kl. So this up to 2, here actually I have given the example of 2 kl. So these cells were grown up to the 2000 liter scale, from the 1 liter to that, and it took around 35 days time for one batch. For one batch from 1 ml to that reaching to the 2 kl is 35 days. After that actually we are infecting these cells. So these are non-infected normal human embryonic kidney cells. These are infected with this Chadox virus, whatever the clone we have developed. Then this virus will start infecting to the cells inside this bioreactor. These are fermenters, or actually we call this as a bioreactor. So we infect to that, and after infection, when the cells will start dying due to this infection of this virus, we will start the harvesting process. In the harvesting, different purification steps are there, in which actually only it will take care that whatever the vaccines we are purifying or producing, so it is only the purified adenovirus. Else actually no any cell debris, the cell debris of the HK cell line or the DNA from the HK cell line, or whether these virus particles are actually if you purify the virus, there should not be the lysed virus particles. So whatever the virus particles we are giving as vaccine, this should be live, because it has to go straight inside our arm, and actually they have to infect one time and they release their DNA inside our body. So it should be live. So for that purpose actually we have designed this process, and this process is taking care of all these things whatever required for the safety of this vaccine. So Serum Institute of India, actually we are already manufacturing. So this is some background of Serum Institute of India. So it is around a 53-year-old company, and we are already supplying around 28 products worldwide in 175 countries, already delivering vaccine. And it is told that actually two among the three, whatever the birth actually or the persons on the planet or Earth, so they get at least one shot of our Serum's manufactured vaccine, because actually that number of different kinds of vaccines, specifically the pediatric vaccines we are manufacturing. And for this project of this Covishield, we have initially tied up with the Oxford, because we have already one collaboration with them. So after that, due to some interest of the UK government, they think that if the Serum manufacturer or Serum get the complete license, so in that case we will not get the vaccine first. So they have given the license to the one AstraZeneca company, that is one Cambridge-based company. So they are also license holders, we are sub-license holder for that. And we have independent, so we have manufactured around 1.9 billion doses. And whatever adult vaccination in India, so around 88% are from the Serum Institute. And from the complete vaccination program, it is around 80% vaccination from the Serum. So to combat on some limitations of the other vaccines, so I have developed this one novel vaccine, so that actually I have not given in detail, but actually that will take years of. So my concept is that vaccine is multivalent vaccine. So it has a spike protein, it has RBD, it has a nucleocapsid. Means at different level it will give the protection to the infection, and that can be used as a nasal vaccine. Means if we give the vaccine through the nasal route, so in that case it has this is adenovirus, whatever the platform I have used just. So on this adenovirus actually there is already expressed the RBD is there, ready-made RBD protein is there. So it will act as a protein vaccine. It will give the immediate response or protection in the respiratory tract. So that is one concept. Then in that actually I have inserted two genes. One is spike gene, which is similar to the Covishield, and additionally nobody has used the nucleocapsid gene for the vaccination. So that also I have used, because that will give the protection before the assembly of viruses. It will inhibit the virus during the replication itself. So that vaccine is also patented, so it is under development, means some evaluation is going on from my side. So for India, these are not relevant to this program, this topic, but for India, nobody before COVID actually nobody aware about the vaccine. So doctors are recommending, so we are giving the vaccines to the free vaccines to our children. Those who are small, 10% people who are aware, so they are buying some branded vaccines and they are giving, because they have money, and there are a lot of branded companies that they are selling vaccines at a huge price out of the reach of these common people. So they are supplying that. So they can buy and they can give. They are somewhat aware. But those who are buying Serum vaccines from the government program, so they are not aware about the vaccine actually. But after COVID actually there is a lot of awareness of biotechnology, vaccine, medical field. Everybody actually increased. So similarly, one unsolved problem for India is the TB. So now the government's target is the TB eradication. So for that we are supporting, we are the key for that. That work already we have started before COVID itself. And now actually the world has, WHO has given 2030 as a limit for the TB eradication. But for India, on the basis of Serum's products, we are targeting 2025 for the TB eradication. And for that actually, main is the detection of this TB. So for detection, the PPD method was there. So from this actually we can detect whether the TB is there. But actually it is unable to detect. It is not giving this false positive results. Also unable to detect the latent form of TB. So for that we have developed the CTB, the kit called, or the product called the CTB. So it is as good as IGRA. So whatever the IGRA test, the blood test used for this TB detection. So it is in human test we have to give the injection to the skin, but it is as good as the lab test called IGRA. And we have given now a name as a SciTB instead of CTB, because we have dedicated that product to the Cyrus Punawala, our founder director, our chairman. And then the detection is there. Then treatment, we have also in the government program with the help of MyLab company, you might have heard about that. So with the help of MyLab, we have designed this program. Means we have given rights to them, and they have took the permission for this antibiotic regimes with their umbrella. So they will, after detection, they will treat the patient. So that will be one act as an ecosystem. If the person is negative for this SciTB detection, so in that case he has given this RBCG vaccine. So this RBCG vaccine, it is called as some kind of magical vaccine. So it will give the protection against the TB. In the initial cases, whatever the BCG vaccines we have took during the childhood, so its immunity lasts up to only the 10 years time. After 10 years, nobody bothered about the BCG vaccination, whether we have protection or not. It is considered that it is a one-time vaccine, no booster dose is required. So in that case, there is increased number of TB patients, because after the adult age actually, after the 18, so there are many cases of exposure to the COVID, and due to our immunity, actually we are not getting the symptoms, but actually we have latent TB exposure already. Around 60% of Indians have the latent TB. So for detection of that, this is the SciTB product is there, and those who are tested positive with the CTB, so they will get 1 month this antibiotic treatment, and if the person is already negative, so in that case he will get the RBC vaccination. And if the person after treatment get negative, the positive person, so in that case also we will give this RBC to them. So that is one program. Another major issue is the cervical cancer. So there is nobody, I am confident that actually nobody get, at least one or two people might get the HPV vaccine from this hall, but I have also doubt on that actually somebody has get the HPV vaccination. So HPV is a severe, cervical cancer is a severe issue in the India. There are a lot of cases after 45 of this HPV infection. So there are different cancerous, non-cancerous HPV circulating to everybody in India. So it is now recommended to take HPV vaccination for male and female up to 45 years age. So there are 3 dose vaccine regime is there. So that we have to take. So whatever the initial vaccines present for this HPV, the Gardasil 4, Gardasil 9. So these vaccines are having a high cost. The Gardasil 4 has 3,000 rupees per dose, so around 10,000 rupees for 3 doses, and for this Gardasil 9 around 24,000 rupees for 3 doses. So these are high cost vaccines, so nobody gets this vaccination. But it will protect the cervical cancer. For that actually we have developed this vaccine. It get the marketing authorization before 2 months. So from this year actually we will start distributing throughout the India, and mostly through the government programs. So it is quadrivalent HPV vaccine. It will give the good protection of all cervical cancers, anal, rectal cancer, and the skin problems of the HPV. So this HPV vaccine awareness and TB program awareness is important. So please tell everybody, they should be aware. This is not for business. This is for the challenge what we will face due to the HPV and TB. So I will acknowledge, because when actually I have a lot of pressure whether I will give good lecture or not. Actually I don't know. So that time, but the head teacher actually requested to deliver the lecture. And thanks ma'am sir for the invitation, the director sir, the Academy of Science, Maharashtra Academy of Sciences, the Vice President Deshpande sir, so for this invitation and guiding in this program. Thank you.
Thank you so much, Dr. Narwade. Actually, yes, is there any question answers? Yes.
There is a growing concern about the post-COVID effects, especially on the neurological systems, psychology and behavioral patterns. So what is the strategy about that regarding diagnosis and then of course therapy?
Actually for neurological, the post-COVID exposure, actually we are not doing anything, frankly speaking. But actually, as per my knowledge, there are no neurological problems or psychological problems due to the COVID, post-COVID effect. There are cardiac problems are there, but not the neurological problems as per my understanding.
So is it a rationalization on the part of the patient that he justifies this as a post-COVID?
Yeah, actually, that is not related. I think the neurological problems post-COVID are not related. The cardiac problems are there. Lot of because this virus itself has ability of to clot the blood. Some actually it interact with some blood factors, and these blood factors leads to the blockage of the heart. Some cardiac problems are there. Yes. Mike.
Actually, diabetic patient, my own sister, younger sister, she had suffered from COVID and she got cured. After diabetes, after that, diabetes is continuously increasing, and now we have to give insulin 4 to 5 times to her.
So she get positive in the second wave?
And second thing, she developed this stroke. So she was admitted in the Viveka hospital. So I wanted to know that is there some remedy to diabetic patients?
So actually, so whatever she has faced problems, so that is due to the steroid treatment which is actually largely used during the second wave. So prednisolone is a key factor for that. So due to that, actually she has a fluctuation in the sugar level, and the stroke problem is due to the infection, because the blood clotting has this virus has the blood clotting abilities, that COVID virus.
Yeah, she had some clot, very small clot in the brain.
Yeah. So due to that, actually
Stroke.
Yeah. So that is the whatever the nature of this infection, COVID infection actually, and it all depends on the body to body variation was there. So that is one of the key.
But
there is
diabetic condition has gone to very worst this. I'm reading a lot about it.
So the steroid is the key. The no correlation of the COVID with the diabetes. The stroke problem, whatever she has, so that is related to the COVID. Yeah.
It has nothing to do with the diabetic, that is stroke.
COVID has a relation with the stroke, and whatever the treatment she received during this COVID, so that is the steroid, so that increases or fluctuates the sugar level.
Thank you.
Most compliments for a very informative lecture.
Thank you, sir.
I would like to go to the beginning of the origin of this pandemic, and its origin is still very confusing, whether it's a leaky from the lab or whether it's from the wet market from Wuhan. So is our technology now so advanced that a lab can actually manufacture a virus which would cause a pandemic?
Yeah, so actually labs can design different kinds of viruses which can cause the pandemic. But as per my knowledge, because there is nothing to blame on the China that actually they have created and the virus was escaped from the laboratory. So this is actually naturally, because if they have designed the virus, there should be some stability in the genome sequences, else whatever the variants after this different phases of the pandemic are evolved. So these are this actually itself suggests that whatever the initial Wuhan variant was there, so these are not lab created. So it is the nature of that virus, so it will get the different mutations and it will infect into the human. So this is the white virus, which is actually they are eating some animals, raw animals. So from that actually they have evolved. So they have one research laboratory was there, virology center was there. So they are working on the different strains of this COVID, SARS, MERS, but from that also they can the virus can escape. But actually it is not lab created or intentionally actually provided to the exposure to human.
And what is the risk that we have taken as a society by bringing down a vaccine development from 7 years to 1.5 years?
No risk to the society, because whatever in the 7 years time, so maximum time is the regulatory permission times. So that regulatory permission time we have actually due to the condition, the pressure from the all the human beings. So actually they have given the immediate approvals. If on the Sunday night we send one letter for approval, so those that letter actually in the normal condition take 1 year's time. So that gets signed on the Sunday night itself. So this is actually the time. And whatever the initial vaccine development experience we have, and we know where actually the risk to the human being. So that factors before releasing batch. So there are different multiple tests, around 28 tests are performed on this virus, each batch lot. So apart from this 28 routine test, I have some different tests, my own testing things are there. So that will show that whether this vaccine is safe to the human, this batch is safe to the human or not. So because that why I have implemented that, because in the first the initial vaccination drive, so there are cases of the clotting, the blood clotting cases were there. So whether this blood clotting was related to our vaccine or it is something different. So for that actually we have tested. So it is not related to the vaccine. So the clotting is related to the COVID exposure. So after vaccination, person can get mild COVID exposure. So he don't get the symptoms from that. But in that case, the virus was there, so that can lead to the clotting. So don't worry, sir. Around in India, around 1.6 1.65 billion people are vaccinated with Covishield. Pediatrics also vaccinated with Covishield, and I am the first who get vaccine in India.
Sir, I just wanted, like you, now we have received majority of us both doses and the precautionary. Like you said, the immunity from the first dose, when it wears off, we should have the second. So what would be the time recommended that after precautionary, what next and when?
So actually after precautionary or the second booster dose, actually we don't have to get another shot of vaccine because we have the memory cells for that. But in the considering the situation, the different variants are coming and some variants are escaping from the these antibodies which are given by this produced from this vaccine. So but when we get the additional precautionary dose, in that case we have already antibody titer, and if we don't take the precautionary dose, in that case after exposure to the exposure to the virus, our immune system will start developing antibody, and between that whatever the maturation, that is 14 days period or the 7 days period for this initial protection, so during that time we may get some symptoms or the severity. So to protect from that, protect from the different variants, we should have active antibody titers. So after precautionary dose also, we have to monitor the antibody titer from our body specific to the spike protein. So that is mandatory. And after vaccination, so there is another question normally actually asked. So our vaccine are developed against the spike protein of Wuhan strain, and then different strains are evolving. So and these are actually data is showing that actually lab data is showing. Lab data means people are taking the antibodies from the human body. They are testing against different variants in the laboratory, and they are showing that these variants are escaping from the immunity. What means escaping from this antibody? So that is one rumor is going on. But our body has some mechanism. So that is called as affinity maturation. Means we have already memory against this virus, and if we get the exposure of the same virus with some mutations in that, so our body will learn to manufacture or develop the antibody against that. And normally, if this is the spike gene, and the spike gene completely we have given as a vaccine. So against it, actually there is no single antibody form against complete spike gene. So antibody is always against small fragments of this spike gene. So this is overlapping antibody. So if the mutation is there, so mutation might be at the small 10% of the region. So 90% is antibody already covering that. So we will not get the severe infection anyway of any variant. Else actually these variants are actually lethal, because in the Omicron the maximum death in the USA are due to the Omicron, where actually we are not getting the normal flu also. So virus, the vaccine is protecting.
So one more.
Yeah.
Initially we were led to believe that CD count in the real-time PCR test, that tells about the severity of the, if the higher CD count, low severity, and then we were told that it doesn't matter, CD count has no significance.
Yeah, CD. So that is CD value. So CD value is that, so it will give the at what temperature the maximum amplification, the CT value, threshold temperature. Yeah. So if the it is calculated up to the 35, but before COVID it is up to the only 30. So if the higher CT value is there, so less exposure of the virus was there. So if the it is less than the 18, so in that case there are some lakh copies of the virus was present inside our swab samples. So it only it is not determination factor for of the severity of the disease. It will only show the high level of viruses inside body. Yeah. If the person is vaccinated, in that case that will also get cleared within some 2 days time.
Any further questions?
Why second wave was detrimental as compared to first one?
So in the second wave, maximum people are at home. They are not exposed to the virus. So that is one first. And the second wave, there was the vaccination drive was started, but actually due to the government policy, these are not for common people. Only the frontline workers they have given. So if the second wave, everybody is get vaccinated, so in that case there might be get good protection. There is low level of mortality in the India. So there is no, means the first wave the Wuhan variant, second wave Delta plus was there. So Delta or Delta plus actually both are same infecting. Means actually it is not like that it is different variant, so actually it is affecting more. So it is only due to the vaccination problem or the exposure problem, because we are also not taking much precautions in the second wave. So after the death rate increasing, we have started some cautiously. Before that, after first wave, everybody is roaming that no, COVID is not nothing, only the business part is there. So, yes, sir.
We have first dose and second dose we have taken, for example, Covishield, and then booster.
It is required that we have to take Covishield, not Covaxin. If there is any specific reason.
Means mixed batch actually, so there are a lot of data on that. So for you if you are talking about the Covishield and Covaxin, in that case, those who received the initial 2 doses of Covaxin and the booster dose of Covishield, they have huge antibody response after the booster dose. So that is already independent study. And those who have received 2 shots of Covishield after Covaxin, they have boosted with the Covaxin. So there was not good boosting with the Covaxin, because we don't know what was there in the vaccine. But the heterologous vaccination is always recommended, because whatever the technology we have used, so it will boost the after precautionary dose also. But if we boost with the another vaccine like the Moderna or Pfizer, so they have used mRNA based vaccines, but actually they don't have the good memory cells from that vaccine. So if they get boosting with the Covishield, so there is a huge antibody response. Similarly, the Covishield after 2 doses of Covishield and the boosting or the precautionary dose with the Covishield also give the good response of this antibody. But when 2 doses of Covishield and 1 dose of Covax, that is the Novavax vaccine, that is spike protein based vaccine, so that will give the highest antibody response as compared to the NA vaccine. So for this mRNA based vaccines, there is a study, so they have 2 doses of the persons have 2 doses of the mRNA vaccines, Pfizer, Moderna, and they have boosted with the Covishield, they have boosted with the Novavax vaccines, and there is a tremendous antibody response. So heterologous antibody in a way preferred for the boosting.
My question may be out of context, but there was quite a buzz in public domain about the efficacy of remdesivir. Everyone was talking about that. And some said that, well, it is not needed at some point of time.
Yeah. So remdesivir was not actually the problem, but whatever the hype was made, the business, the malpractices, means actually the fraud remdesivir from the unauthorized sources are coming in the market. So that was a problem for that. So remdesivir the initially used for the Ebola treatment. So it is specific for the RDRP genes. It inhibits the RNA-dependent RNA polymerase of this virus, for any virus, Ebola and these COVID viruses. Just actually, we don't understand when which remdesivir injections and how much we have to give to the person. So that actually leads to the other problems like sugar problem, because we are giving the immunosuppressant, then the antiviral through the IV, so that is some problematic. Thank you.
Thank you, Dr. Narwade, and we wish Serum Institute and your team all the best for the future vaccines that you may be coming up with. We are looking forward to nasal multivalent vaccine and the recombinant BCG. Hopefully 10 years from now, children will just be required to take 1 RBCG when they are born, which will protect them against all the diseases under the sun. So let's hope for that day. And thank you once again to the Serum Institute for all the hard work their team has put in. May I request Dr. Vidula Kapre to please hand over a memento to Dr. Narwade as a token of our deepest appreciation. And may I also request Dr. Kapre to present the vote of thanks.
So that token was really a token. I don't think sufficient enough to express our gratitude for the wonderful, enlightening lecture on novel strategies for COVID. Thank you very much, Dr. Narwade. It was very a learning experience for all of us. And as you rightly said, 2 years back, most of these terminologies we had not even heard in our lives. Similarly, 2 years back, when we were suddenly struck with this COVID pandemic, who would have imagined that a day will come when we'll have a hall packed with audience and together we will be having this lecture. So I take this opportunity to pay tribute to all the scientists who really hit the ground running. As soon as we were faced with this pandemic, I think they immediately started working on understanding the virus and finding out ways to counter it. What we heard from Narwade just now, I think, was just the tip of the iceberg as regards to what all efforts, untiring, research, technology and whatnot has gone into the making of this vaccine. So really, hats off to all the scientists here, and special thanks to Dr. Narwade for at least 2 reasons. Number one, of course, as a leader in the development of this Covishield vaccine, which, as you all know, has proved as a savior for humanity. But also equally important, it has put India on the world map as a front runner for developing a safe and efficient vaccine, efficacious vaccine in such a short record time. So that is a big contribution, and as Indians we should really be proud about that. The second reason is that Dr. Narwade, being so busy, he's always shuttling, I think, between Pune and USA, that he has taken time out to come to Nagpur and to deliver this beautiful lecture to us. He only arrived this afternoon at Nagpur and he'll be leaving immediately tonight. It will take him into Pune at an unearthly hour of 1 a.m. and I'm sure he has a whole list of some important engagements lined up for tomorrow. So on behalf of all of us, I sincerely thank Dr. Narwade from the bottom of my heart. VNIT for last several years is a co-host for this event and we are deeply indebted for that. But that we have resumed this lecture series, the credit entirely goes to the director of VNIT, Dr. Padore sir. Because when the pandemic put a spanner in our program, I kind of lost momentum and I went into that thought process that maybe time has come to put a full stop to this program. And it was Dr. Padore who motivated me to continue. Today he had some very important engagement, inauguration of a health center at VNIT, for which director of MUHS is here, and that is why he had to leave early. But he kept his promise, he was here with us and he shared his valuable thoughts in his presidential address. So I want to take this opportunity to express my gratitude towards Director of VNIT, Dr. Padore sir. Dr. V.K.Deshpande, Head of Physics Department, VNIT, Vice President, Maharashtra Academy of Sciences, plays multiple roles for this program. He represents the Maharashtra Academy, he takes care of all the arrangements at VNIT, most important, making this hall available for us, and for spreading word around in the scientific community about this lecture. Thank you very much, sir. It is said that all the beads in the world cannot make a necklace unless they are tied together. Over the years, all organizations and individuals who contribute for this lecture are definitely tied together through a common motivation. This year, a new bead is woven into the necklace and is in fact shining like a diamond. Dr. Rashmi Uddanwadekar, Professor, Mechanical Engineering, Associate Dean, Public Relations, VNIT, She has joined our organizing committee this year itself and has already proven to be a big asset. Thank you, madam, for everything and special efforts taken to publicize this event. I want to thank both the print and electronic media for covering the program. Department of Physics, VNIT, under the guidance of Professor Deshpande, is a big help with the audio visuals and other arrangements here, and my sincere thanks to them. Thank you, Dr. Mangulkar, for support in photography. My staff here from Niti Clinics is a big support system, and they all work for this event as their own. Thank you very much. I will not take names in the interest of time. I know my family is emotionally vested in this lecture series because it is in the memory of my father. But I take this opportunity to acknowledge their unflinching love and support. Sorry. I lost my mother in February this year, and she used to take keen interest in this lecture series. She would attend each and every lecture, even the last one, in spite of her advanced age, which the last lecture we held in 2019. So we do miss her, but I'm sure wherever she is up there, she must be happy that the show is going on. Our pillars of strength, I have saved them for the last. Professor Apte sir, Singru sir, Sapre sir, always provide valuable guidance and inspiration. Sapre sir is in UK right now. Singru sir, thank you for coming in spite of other family commitments. Apte sir, you are like my go-to person for any advice related to this program. And as Gauri mentioned in the beginning of the lecture, the importance of 8th August in the history of science, which Apte sir very diligently he found it out and handed it over to us so we could share with you all. So 8th August is not just the birthday of Professor Bhide, it has many more important things attached to it. And to this effect, I want to say that as much as this lecture is in the memory of my father, it is more about his principles, his ideals, the constant endeavor to better the cause of science and to popularize science among the masses. So the real success of this lecture lies with you all, the audience. It is your participation, it is the interest shown by you all that helps us achieve what we strive for. Thank you very much for coming, and please continue to shower your affection, and let us continue with this lecture series for years to come with renewed vigor. Thank you very much, all of you. And please join us for tea.